Well, good morning. I'm Tom Gardner. It's really wonderful to have you all here. So the Maritime More Vision Initiative is an idea. It's an inspiration. It really sprung from the heart of Robert and Mary, and now it's infected all of us. Fortunately, right? But it's still just an idea. So in order to get this idea into reality, which is what we only care about, we need a legal and financial vehicle. So that's called a consortium. And there have been many consortium. And so what that means is a structure that will be housed within the University of Michigan as the honest broker that will be able to make contracts, to receive funds, to disperse funds, and to really keep us all working together. You just heard a great example about the Ocular Biorepository and Resource Center. It isn't just a repository collecting tissue. It's a resource center to get data and tissues from Patrice's lab into the hands of academic and industry and other people who want to analyze it and use it. So it's really an exchange. And so the partners will have a chance to contribute their knowledge, their insights, their questions, and also to get samples, protein samples, tissue biopsies, tissue data, and to take it in their own shop. So the whole idea is that this is a pre-competitive consortium. And Matthias is going to talk to us more about his. But the idea is that by having this as a vehicle, then we can all work together. It also extends to the clinical studies that Jenny has discussed with you. And that means that the partners, so if anybody who is really interested in participating in this study, come on, let's join the team. And by the same token, the partners can take that information and they can take it into their own shop. And if they find that there's a target from the clinical studies or from the tissue studies, then they can patent that and develop their own product and hopefully get that into the hands of patients. So at any rate, we are very fortunate to be able to have Dr. Matthias Kretzler with us somewhere. Yes, here we go. Matthias is a professor of nephrology extraordinaire here at the University of Michigan and leads the Kidney Translational Research Program, which is, like NPOD, has been very formulative in generating the ideas for the initiative. Matthias, thank you.
Good morning, everybody, and wow, what firepower you have assembled in this room. Congratulations. This is really, really impressive. And I would like to mention to the patients that, yes, you should expect that this consortium will impact the lives of people with diabetic eye disease in the very near future. And as you saw already, there are success stories you can build from. So what Tom asked me to do is to give you a quick overview about consortia and framework we have developed in the diabetic complication framework we are working on. Yes, I'm a nephrologist, but with Tom, I'm co-leading the JDIF Center of Excellence here at the University of Michigan, and we are taking care of all organs impacted by T1D, from pancreas to kidney, nerve, and eye. And you will see the presentations of the strategies we are deploying are germane, and also the frameworks to bring public-private partnerships together. And this is a place where I really want to give a great shout out to Fast Forward here at the University of Michigan, where we did find very open, creative solutions to bring industry and academia together for the good of our patients and obviously for all players involved. I want to start out with a concept which I think is important also for the patients in the room to understand. Yes, there are lots of players which we identify right now, that they work in concert. Gene and environment work together. because people with the same glycemic control will have very different damages of their retinas, and vice versa. Even with very variable degrees of glycemic control, you can have similar disease phenotypes present. And the fascinating piece is, as we heard, we have learned a lot about genetic determinants and environmental factors who are the disease-driving initiation events. But the fascinating piece is that those are driving forward the disease progression and channeling together in a few pathways. VEGF obviously is the main example in retinopathy, which has established at least some therapeutic efficacy in the States. And this supersedes a lot of our conventional disease classification if you look into that. And that slide can be used for kidney or eye or for nerve or for any chronic progressive diseases. And the beauty is, and this is for the pharma people in the room, that with this knowledge in place, we now can identify where and which molecular pathway is active in a given individual and where and how to intervene and to bring efficacy to our patients with chronic diseases. And this is the time of genome scale profiling. Yes, this is a lot of data, and the art, and we will discuss it in a moment, is to transform the data into knowledge in each and every one of your knowledge background you have. And that is as difficult as generating the data in the first place. So we have spoken a lot about different aspects, how to generate patient-level information. In the kidney field, we have the opportunity to use research biopsies regularly, and we are doing that broadly across many networks and trials now around the world to give us tissue-level information. But as you heard from Patrice, that's a reality for the eye field now as well, with different approaches, but with similar dense data sets. Blood and in the kidney field, urine are obviously valuable biomarkers. What I have not heard much is the fascinating capabilities we are using in the Center of Excellence now extensively, that we obtained inducible pluripotent stem cells from study participants. We obtain all sorts of other parameters, including often a kidney biopsy, then to benchmark the organoids as we differentiate them out into kidney, nerve, retinal component, to see where that ex vivo system, the model, is matching the human. And then we generate, obviously, a plethora of information which, with our systems biologists in the teams, have learned how to connect their data together. And with the cross-cutting disease mechanisms defined, we identify factors who are associated with long-term loss of function. And the exciting piece now, we can bring that back to the individual patient level for patient-level pathway assessment. And that's a reality in 2023. This is not a hypothetical concept. Let me show you how we have made that a reality. That figure is not a coincidence that you find that in your booklet, as we closely collaborated in building our research networks jointly up together, where you can see the multiple elements aligned. And the key piece is the loop coming back, that you can see that clinical trials now can provide us information which mode of actions work and doesn't work. And that, I think, is a key lesson also for pharma to learn where are the blind spots of the current therapy, where additional interventions will have maximum impact for the unmet need of our people. I want to give you one example out of the Kidney Precision Medicine Project. This is an NIDDK-funded large research effort where we took this concept forward for people with diabetic kidney disease. As you know, they are not routinely biopsied. In KPMP, we obtain research biopsies from our study participants. We then use centers of excellence around the nation to generate the multiscalar data set in spatial organismal structural resolution of the kidney and push it forward with a real definition of targeted therapies and the framework of continuous discovery and evolvement. The concept is aligned with very much what you heard from Patrice and NPOT, that we have excellent centers to recruit samples after careful training and auditing with very rich metadata. As you also heard, if you generate large-scale data set, the metadata, the associated information, where and how samples are coming from and how they were processed, is as important as the data itself. We are using the same CBR here in Michigan, Vicky Blank's operation, to assemble the material, to distribute it then across our tissue interrogation sites, who generate the multiscalar data sets which are integrated and then shared with the kidney tissue atlas. Yes, this is a complex organization, but clearly you have the expertise to manage that in the frameworks and the people you are working with here. That the central coordination unit with the CBR here clearly has all the quality standards in place to adjudicate, track, monitor, and ensure sample integrity going forward. A critical piece, I'm very excited to see that really integrated in an integral part of what MTM is doing here, is really the patient and central role in the development, in the execution of the effort. And as you can see here in our studies, we have embedded patient not only as participants, but actually as true leader in every step of the process. And it's nothing more grounding that if you have a patient panel, when you start your effort and when scientists get into debates which might not be always productive, a patient telling them, look, you are here to make a difference to our life. And the sobering aspect, certainly with diabetic kidney disease, is that yes, we started with 12 patient representatives, and we have now 9 still active in the field because we failed 3 of them. So this is a very effective way to keep everybody honest and on focus. Quick overview, which you have already seen a little bit in Patrick's representation. Really, please, as you build that research, reach out to centers of excellence who have rapidly evolving, enhancing analytical capabilities. You have a significant unmet need. You have resources available, the technologies are evolving. And we were able in the kidney field to actually have the technologies evolve so they can utilize effectively the way we can procure samples from patients. Yeah, it's not that we try to make the sample procurement fit technologies, rather we have excellent sample procurement protocols in place, and then the technology are optimized, adapt, using similar like you do in the eye, post-mortem material to actually optimize the technologies before you allow them to access your relevant samples. And this is really important, and this is painful, and academic scientists need to be educated that as important our data is a quality control tag to the data at every step. And our data density is as intense with these metadata, the attached data types, as it is with the data itself. We have published extensively on the topic, all of our protocols, ensuring these elements are publicly available in Protocol IO. So feel free to utilize them to whatever extent possible. And if you learn lessons, please let us know them as well. The key next step, and this is really an essential feature, and we are very fortunate to have Nikki Bonovich, the director of MyKTMC Software Solutions, in the room here, so feel free to talk to her during the break. How then you build a data sharing and mining ecosystem? Yeah? Because large-scale data sets are useless if they are rotting in your drawer. You have to find out what each and every of your user needs, and this is different. We are sharing our information back to our patient participants in a very different way than we are serving it to the expert bioinformatic data user. And you can see here the 6 exemplary user persona we identified early in the project and did extensive user interviews before we started to map and build out. You need to build the system so that you answer not the question you think you want to answer, but the questions your user want to answer. And the people in the room is a fantastic audience to do that, particularly the industry users. And we just had an outstanding KPMP industry roundtable at our annual meeting last week, where we heard from industry that we succeeded with that our data sets now are integrated in each and every of the large pharma pipelines. And if you don't have human validation of your data coming out of the research networks from RPC Square and KPMP, you're not going anywhere. And this is fantastic to see that this worked. So again, please feel free to leverage how we did. This was a lot of groundwork. starting with the data sources. We build an ontology framework, how to mapping these different data types onto each other, and then develop different data exploration tools. Some of them were developed in-house. Many of them were co-developed with the Human Cell Atlas and the HubMap program. If you want to explore it, go to the website. It's open data, open science. You can work with the data. You can download the data. If some of the information is relevant for you, like endothelial cell stress factors, we have 12 different endothelial cell types mapped in KPMP now. Many of them might be of relevance also to retinal epithelial cells. We have raw data, openly accessible, only if they contain sequence information, potentially identifiable individuals. We have a single click through the DUA to do that. We spend a lot of time to develop an explorer tool who brings in molecular and cell biologists without deep bioinformatic expertise. And we, using the spatial molecular mapping revolution currently taking place, have built with Niels Gehlenberg from HubMap Harvard an advanced Atlas spatial viewer tool, bringing spatial metabolomics, proteomics, and transcriptomic data sets into the Atlas. And that building the community has worked out in a way that we shared our protocols and strategies early and often. And as you can see, we have global access to the database with over 20,000 downloads. These are not clicks. These are downloads of data who are embedded. And it's exciting to see at the last annual meeting 10 days ago that a third of the posters using human data somehow interacted with the data resources we had generated. One example, just to give you a flavor of where this is going, this is a paper we published in Nature a couple of months ago together with the Human Cell Atlas and the HubMap Consortium, where we took our kidney data sets. We used two different cell level annotation, single cell and single nuke sequencing, then followed with the epigenetic attack sequence data to group samples from kidneys together. We identified over 100 different cellular states in health and disease, and then map these states together in their 2- and 3-dimensional context to identify not only how cells are transitioning from a healthy to a stressed to a regenerating, recovering, or progressing stage, but also how they talk to each other. And you saw from in Patrice's presentations already that these strategies are clicking in the retina now as well. This is exceptionally powerful data for the drug hunters, because now you can see which cell is talking to which cell in which language, and what is a happy neighborhood and what's an unhappy neighborhood, and how you can make the neighbors talk decently to each other so that you transfer your stressed neighborhoods back into a reference state. Also, what Mike has shown to you, it's critical that you have already data available with long-term outcomes. So you can, like we did here with our rare disease network, map these deep data sets from the current data back into structures, informations where you have the long-term disease strategies available. The patients ask us, okay, this is a lot of data and maybe some knowledge, but are you making a difference? And the answer is yes, we do. This is the rare disease space. I'm leading the NCAT's rare disease network for glomerular diseases, where we took that knowledge network I have described for you, which we are now establishing for diabetes disease. Here we have 14 years of work behind that, where we now can take the knowledge network and we identify in our heterogeneous group of patients molecular profiles active in each of these patients, going from tissue level information down into biomarkers non-invasively captured from the same patients at the same time, so that we can link what's happening in the tissue, in the endothelial cell stress, in the epithelial cell compartment to blood and urine. We monitor our patients, we communicate the concept which pathway is active in a given patient, and which trial is targeting the pathways which are currently actively destroying your organ function, and then bring these people to the clinical trials which are active in parallel. Heather Asghani, who is in the room, has established a public-private partnership with NIH behind that, so that we have a level playing field where now 6 different trials are playing with our patient pool, and we are assigning the right patients to the right trials where everybody benefits. The trialist gets more homogeneous patient population, can detect effect sizes with smaller samples, and our patients are more likely to get a drug trial exposure, which might actually be beneficial to them. this is with trials. How can you take that forward into treatment care and treatment care stratification? This is an effort at different public-private partnership. In Europe, the PRIME CKD Consortium, led by Guido Lamberspinker out of Groningen. It emerged from the BDKD Consortium. Marlon from ADA was one of our lead scientists in some of his former roles involved, where we established initially a diagnostic biomarker panel to identify the right drug for the right patient. But more importantly, our patients also told us, but please, less drug for all, meaning when do you know when a drug is working and when do you know when a drug is not working? And this is the same framework I have shown you for the rare disease, now for diabetic kidney disease, with an additional critical element that we are talking both to regulators to identify effective paths forward for outcomes and treatment response biomarkers to be linked to these outcomes. And please don't forget also to pay us in ways to actually use the biomarker profiles to enrich for patients more likely to respond and thereby justifying significant therapeutic investments. So how to bring all these players together onto a common stage, and as I mentioned earlier, public-private partnerships are a key strategy here, and the genome scale data make them so much more easier to establish, because on the lower panel of the slide you see the knowledge networks, which I have described for you in the 3 different settings before, and those are a key element for both academic and industry researchers together to go on the drug hunt. And you can learn, and the questions we were asked early on before we had the partnership were always the same. So we told then our different partners in the bilateral context, come on, let's get together. If you all need carefully annotated human data sets with long-term outcomes linked to it, let's generate them genome scale, and we have them on a common table. We design and develop next rounds of study, that's the top flow diagram above that, where we jointly together in the steering committee identify these are key missing data we need to have, like 3 years ago, we are seeing effective therapeutic trials. We need tissue, we need biosamples from patients responding and not responding to the treatments being laid on the table, so that when you are looking at a target behind your firewalls, with the data you have ingested in your pipelines, with the data mining tools we have developed jointly together, you know if your target is or is not adequately addressed by the therapy which is becoming standard of care. If you want to have follow-up discussions, Dr. Ascani will be available over lunch to brainstorm how these things can be worked and the many nitty-gritty details necessary under the hood to make them work. But the consortium is in its 9th year of funding, and we just recruited 2 new partners to the consortium. So this is an effective model to bring academia and industry together. With this, I would like to wrap up. I hope I have given you a flavor for how different questions, different interactions in different frameworks can work together. And by finding creative ways to align interests, we all can be successful to keep our people with effective organ functions in diabetes. Finally, I have to acknowledge the KPMP network. We have over 200 investigators across the United States and Europe. The Rare Disease Network for glomerular disease, my team here in Michigan, who makes all that work fun, and most of all our patients who are eagerly awaiting for us to combine our forces together. Thanks for inviting me to give you the overview of our approach.
Okay, so in continuing with the theme of keeping patients front and center, we have Carol Oxenreiter here, who's a person with diabetes and is very long experience with JDRF and their work. So Carol, can you give us a little thoughts, some of your thoughts about the notion of a consortium for complications like this?
Thanks, Tom. Yes, as Tom just mentioned, I am a lay person. So when I think of consortia, I think of what Dr. Kretzler was saying earlier about making your neighbors get along with one another. And I think it's really crucial when you look at this disease and how there are so many disparate parts of it, that it is imperative that we get consortia so that people can work together to try and make this move faster. It's been really interesting just being in the room today, listening to people like Dr. Chang saying he's learning new things from people. Dr. King is referencing people that he's learning things from. That's the whole idea for me of why you have to have a consortia, is to get people to talk to one another and find things that they don't know and admit they don't know so that they can learn from them. We're lucky to have Dr. Atkinson here today. Anyone who's ever attended an NPOD annual meeting, the best part of it is when someone raises a question and challenges someone else on what they've just said they've learned, and a robust discussion occurs. And it's a learning opportunity for everyone there. When you have business in the room, when you have academia in the room, when you have patients in the room, you're afforded the ability to hear different perspectives that enlighten everyone. And for me, that's the key to why these things work and why they're so important.
Great. Well, thank you very much, Carol. So there is a very active ophthalmology-based consortium in Europe called MacuStar for the study of macular degeneration, and Dr. Ulrich Luhmann from Roche has been very active in that. So, Ulrich, can you tell us a little bit about your experiences, both some of the good parts and some of the challenges that perhaps you've seen?
So thank you for the opportunity to contribute to this discussion. It's great to be here and share that view. I think we had heard a couple of very important points in the talk we just learned, and that resonated a lot with my own experiences in ophthalmology. The first thing I want to say, these consortia can work in ophthalmology, which is great. So it's really good to see that in a pre-competitive space, the big competing companies, usually on the drug development side, can come together and work really closely together. But one of the key prerequisites for that is really this equal level playing field. So you really come together with key experts from academia, the pharma companies come together, but also it's clear to be very openly developing a kind of trust to address these questions and respect each other's perceptions and views on questions and how we see things differently, and accept that needs to come together to be successful and bring things forward. So I think there's a lot of resonance here that is important. And I think the key background for everyone who's not knowing that, in that consortium we have 3 big pharma companies, Bayer, Novartis, and Roche involved in ZEISS, and 7 big academic centers leading this, and then a clinical trial is being run for now, going to be a 6 years follow-up trial in intermediate AMD, very much a similar thing here. So we need to really define the common objective of that. What do we want to achieve with it? And it was very clear with that concept in mind, a common goal to define that and not have these divided interests and maybe academically maybe thinking, oh, we want to explore the disease mechanism very broadly, omics, and why would we do that? And then samples go in that direction, while pharma is very diversely running in a different direction. No, we need this kind of short-term follow-up clinical biomarkers which help us to validate things. So that was a core initial discussion to bring things together in that space. So I think that's a good thing. The challenging thing is really to keep, from a business perspective, the interest high enough for these kind of mid and long-term investments, because decision cycles are short, interest shift within pharma companies, so it's really important that these kind of models, I would say, are really highlighting the cost effectiveness aspect and how we serve that common goal most effectively and have our investments from all ends, academia and pharma, really come together to a common goal, which is mid to long term. And having good argumentations around that and discussion around that, how we all benefit from that in our different aspects, are really important. That's a big challenge to come together to that.
Well, we appreciate all your experience at that. So we have another representative from pharma here, Dr. Phil Lai with Perfused Therapeutics. And so Phil, perhaps you could tell us a little bit about how Perfuse looks at the prospect of being part of this consortium.
Thank you very much for the invitation and for this amazing initiative to speak here today. I'm going to speak from my opinion. I've been both in large pharma and small biotech, and I think there's obvious synergy and the whole is greater than the sum of the parts when you participate in a consortium. For what I'm taking away from this, from a pharma drug development perspective, is that what can pharma do to bring value to the consortium, and what the value does it get out of it, to Ulrich's point, so that they stay involved. And one of the obvious things is just direct resources, right? So pharma does have resources that can allocate to these kind of endeavors. And at the same time, they can provide some special technical expertise that all the different companies might have within themselves. But they could also provide resources and develop areas that they don't have special interest in or special expertise in, to help and work on areas that they can not allocate resources to within the company, but can develop within the consortium. So number one, just resources. But number two, I think what's really striking to me is the need for harmonization. It's been brought up multiple times now today. You know, all the industries running different clinical trials with different ways of coding the data, of different ways of capturing the imaging, different ways of analyzing. And I think to be successful as a consortium and to try to bring all this data in together, to have open data source that can then correlate with the biotissue repository, correlate with the PROs, is the real need for harmonization. And I think pharma really needs to step up and work together to get a standard coding resource, a standard common data, so they can facilitate acquisition, exchange, and submission of data, both in just the clinical trial patient data as well as the imaging data, standard protocols for capturing imaging, standard protocols for how you even do BCVA. You know, I think even across trials I've seen at Genentech, different trials have captured different methodology, how to capture ETDRS. And so all these different kind of disparate ways of capturing data need to be harmonized so that we can collectively use this in an open database to be able to facilitate better clinical trials, better subsets of populations, and then just kind of improve the ability to run the trial quicker and more efficiently.
Fantastic. Well, thank you very much, Phil. So next, we have Mr. Drew Woos, Dr. Drew Woos.
I wish. I'm probably the only non-doctor here.
Okay, that's all right. That's why you're here. And but your work is really, your business is harmonizing data across large data sets. So you can talk to us about that, which is obviously a common theme here this morning.
Yes, absolutely. Thank you, and thank you for having me. So my company is a professional services firm, and where you guys typically call me is after you've decided to make a very ambitious data project, and you say, hey, Drew, you know, the DRCR, for example, the network wants to follow 400 patients for 5 years longitudinally, and we're going to image them all, and I want you to grade them, but we need to, you know, send them over and have automated analysis of all these images, and that's a piece of cake, right? And that's where we come in, and what I hope to really help you all understand are that there's a lot of intricacies and a lot of detail, and a lot of my red hair has gone away trying to create data sets that have the quality and the ability to be run against new models in the AI and be trained and labeled very, very effectively. And so I could go on for hours about individual stories of weird images and problems, but what I would impress upon people as you think about building the consortium is try and think of the initial folks, the people behind the camera, the people collecting the data, and try and make things as straightforward and easy and repeatable and clean for them as we possibly can, so that you can collect more data more efficiently, cost less money, less cleaning, and it's just a better overall outcome for your data, which we've learned extensively with the network. They've done lots of work there. In my other practices, we work with the American Indian and Native Alaskans for teleretinal screening for diabetic retinopathy, the 100 clinics, and when you don't have patients every day, or you don't have your clinicians needing to do the same data collection day in, day out, it gets difficult, and you want to make sure that we can collect this information very, very cleanly. So that's sort of point number one. Point number two and I will not point out any particular clinic, please don't send me iPhone screenshots of your image review software. Anyway, I said I could go on, I won't. But there's a few things that I would like to see a group like this try and accomplish with where you're going. And one of them is an ability to create, and for me, what would be a retinal imaging data set that's longitudinal in a way that it can actually follow patients between clinics as they move around, as they exist in the world. And we have a lot of real-world data collection that we do. And I'd like to see us think about this in terms of almost like a master retinal index or a master patient index of images for people over as long a time as we can. And when you're looking at things like the biobank and whatnot, can we go backwards? Can we see the prior 10 years before that sample was donated? Can we get that information cleanly and accurately into something comparable, something that we can train the AIs and ultimately lots of algorithms and come to different little pieces that you can string together to help collect the images well and cleanly and ultimately have a great outcome for your patients.
Well, thank you. You have a tremendous amount of experience. Next, I'm going to call on Dr. McLeod. Stephen, you, as former chair of a large, complex department and now the CEO of the American Academy of Ophthalmology, you're responsible for all of ophthalmology across the whole world.
Thank you.
But no pressure. So, and central to that is your ability to get people to talk to each other, right? So tell us about how the lessons you've learned, please, that would apply to putting together a consortium like this and having it successful.
Yeah, no, absolutely. That's one of the real challenges for the American Academy of Ophthalmology, because we really do lie at the nexus of patient care from the physician perspective, from the patient experience, dealing with academia, dealing with the federal agencies, and really trying to pull everything together with one point in mind, which is what is in the best interest of the patient. The challenge, of course, in this particular sphere is that we are dealing with incredibly expensive processes, generating a biobank, generating the data, generating the insights that are going to move us forward. And, it's sort of the old adage that, what is the most expensive department that you can have in an academic institution? It's not mathematics, your second cheapest department. It's going to be, your biomedical sciences. The big challenge that we have with the consortia is, it takes huge resources to put them together, and then the real goal is to try to get all of the information out to everyone as quickly and as broadly as possible. But how do you balance the costs that it takes to generate, to curate, and to distribute, with the fact that, at the end of the day, for it to get out as broadly as possible, there are a lot of very valid users who, don't have the means to actually contribute to the generation and the management and the curation of the data. And so, from our perspective, really one of the things is to recognize that reality and try to figure out and to mediate, what are the criteria that help to stage and sequence the access to the data. How do you try to parse it out so that it actually can get out to individuals? And how do you manage that with the incentives that you need to have for people to participate in the consortium? All of these are really very challenging, but at the end of the day, I think that we've now really come to the point that we recognize that it's this sort of pooled, aggregated cooperation that is absolutely necessary in order to move our fields forward, and we just have to be creative in trying to solve the problems. I really was really fascinated with the approach that Matthias described, because it seems as if you've managed to get to the place that you're very comfortable and able to operationalize a true open access approach, which is really quite an accomplishment.
Well, thank you very much, Stephen. So all this, Shelby, all this boils down to you, since you're running FFMI and representing the University of Michigan discussion with the companies. And can you tell us a little bit about the process you go through, some of the what you're looking for in terms of these relationships? Please.
Yeah, absolutely. So I think the easy answer is the purpose, right? We all are here today because we agree on the purpose. But when it comes to academic and industry partners, the devil's in the details. And I think any of you in this room who have endured execution of a contract between an academic and an industry partner knows that it can sometimes be a long and painful process. But we're really, to our benefit, we are able to draw on the experience that Dr. Kretzler has in putting together this type of consortium in the past. So we can learn a lot from those experiences. But I think as we move forward and navigating what these relationships look like, who's bringing what value, and how we can de-risk these large investments to build these resources to achieve our ultimate outcomes, is really to keep that purpose in mind from both sides to smooth the process of negotiations and getting everyone on the same page.
Great. Well, you do a great job at it. So you talked about the investments, and that's really where the crux of the issue in many respects. Maybe I'd ask you and Matthias to talk a little bit about the scope of the investment to get to really a functioning consortium over a period of, say, 3 to 5 years.
Do you want to start?
I think the beauty is if you would start from scratch significantly, but you don't. And that, I think, is a key asset. That when we initiated the Renal Pre-Competitive Consortium, that was leveraging 21 years of very hard work from 3 networks already, where we had data already in the open space, which we could easily ingest. We had people with deep knowledge of these data, and that is the real asset. Data, yes, but people understanding the data is the next step. And the most critical step is people understanding how to communicate with the different stakeholders around the table. And that, honestly, I feel is our strongest value proposition. That we have data, many of them are now in the public domain, so no, you would not have to join us. But together we identify how to use their data, we identify the key gaps you, the drug hunters, have to utilize their data to make your key decisions, to go up to your boards and to defend their target and to get that approval that you go into the clinic or that you go into phase 3. And that is a unique asset, understanding in the academic side the challenges, and we call our patients, our participants, heroes. I call the people on the drug pharma side different heroes, because the battles you are fighting are very different and were unbeknown to many of us on the academic side, but are absolutely critical for us to win that war. And for academic and the network to understand what you need and then jointly develop solutions for that is key. And then the individual investment, depending on the size of the consortium, can be very, very modest. Certainly in compared to what you have to invest into even a phase 2A study, it's very modest. But it can allow you to leverage and to stratify. And as I showed with the Rare Disease Network, we are cutting down trials from 384 to 62 patients with this approach. And that in itself, recoups the investment of the entire nephrotic syndrome study network from the NIH and Michigan over the last 14 years.
Fantastic. Well, how about this is a good time to seek some questions from those of you sitting out there? Or comments?
Sure. I have a question for biorepository. I think that I hear a lot of data collection that is going to be the vitreous, the donor cells, donor tissues and such. What about the aqueous humor? I think especially Phil and Ulrich from industry is here. Maybe you can comment on how useful it is to collect aqueous humor samples from the patients and for biomarker development or target discovery.
Does Patrice or Jeff Sundstrom or anybody else would like to comment?
would be happy to comment.
Okay, please go ahead, Ulrich. Yep.
Some internal efforts on that. I would be happy to comment on some of that. So we are using actually aqueous humor in that space at the moment to really explore the possibilities and evaluate the value of that matrix. We all appreciate the potential limitations of that, being it really in the anterior chamber. So what the key questions we're trying to answer, first of all, is it relevant to monitor some effects from back of the eye? How do we do that? We need to link it up with matched sampling, aqueous humor, vitreous humor, and potentially some efforts we have heard here today. So it comes back to a common theme we have heard prior in the discussion already. Matched sampling at a time where we can really understand relationships between clinically accessible samples doing trials, which are not our biopsies, which are not usually vitreous, but potentially aqueous, which has its challenges, but it's being used in trials currently to answer questions like disease-related monitoring, trying to link imaging biomarkers up with molecular, really high omics-like data sets from very small volumes. So really that's a very attractive path, and it looks relatively promising in that space, that there are certain use cases for that in drug development as well. So I really shout out for matched sampling in all types of samples in the patient population.
Very good.
Just to comment on that, in the kidney field, 8-10 people were fully sufficient for us to bridge. So that if you have a pathway mapped out in a system where you get deep access to the organ, and then you don't do a genome scale mapping for which you need hundreds, if not thousands of cells, but you just need then to map what is the correlation between my tissue level definition and a fluid biomarker. Then if you have good variability, that bridging can be done with a very small sample size, and you find patient volunteers who are willing to do that if you ask. Yeah, you really do. That's one of the surprising facts, again, we were able to bring to our community, that if you ask, patients will volunteer, particularly if they have experienced in some of their loved ones what the disease can do to them. And then you can flip over and you have the non-invasive surrogate, and you can go across, ideally in some retrospective studies, some clinical trials and cohorts.
Thank you. Patrice?
I'll just echo what Ulrich said. The key at this point is just how much does the aqueous humor reflects what's happening in the back of the eye, and I think we have very little knowledge at this point relative to this. So it's going to be something to be demonstrated. There's certainly a lot of interest. I completely understand that. In terms of the biorepository, that's not an emphasis that we have made yet. We've decided to start with correlation retina vitreous that we've identified was also absolutely missing, and so we're doing this right now. But that's perfectly clear that it's also a level of correlation that we need to learn about.
Thank you. Any other questions? Ramin?
Yeah, hi. This is Amita here from the Wisconsin Reading Center, and going not about the aqueous samples, but for imaging purposes, for data sets, talking about what Matthias just mentioned, that they started off with 20 years of data that was already existing. I think in diabetic retinopathy, we do have significant imaging and other phenotype data that has been curated already that we forget about. Yes, there are all the industry trials, and it's very hard to get those kind of imaging into open source and things like that. But we do have a lot of NIH studies that have been done for 20, 25 years, the EDIC study, the DCCT, DPPOS, that have beautiful curated data sets that are available, natural history, that I think if there's a way to bring those out into use for people rather than start a brand new trial and recruit, those are plenty of studies that are available out there. ACCORD, for example, 8 years of follow-up on cardiovascular risk factors and eye diseases. There's just a plethora of information.
Well, that's a great point. Thank you very much for keeping that in front of us. And we're certainly open to utilizing that as much as possible. Thank you. Ramin?
One of the side effects of internet companies using data of people for different uses is that there is more and more strict regulation, at least in some countries in Europe, for use of data, including data of patients, including biologic samples, asking to have the individual authorization of patients for the use you want to do, which makes it really complicated because when you have the data set, you don't know how you use it. How do you manage it each year? Is the ethical committee authorization is sufficient to use and to do whatever we want with the data? Or how we will manage it in the future?
I think there are many different ways how to approach it. I can give you 2 examples. Obviously, rigorous consenting is required, and I think actually adequate under giving the fact what deep knowledge we are gaining from study participants. Here at the University of Michigan, we are GDPR compliant, so we are reference resource centers for many studies who are recruiting out of Europe and sending data and samples here. So that base is covered. It was not a small feat to make Michigan agree to a few of these things, but we did it. And the other piece is data not necessarily has to travel nowadays any longer. In the KPMP framework, we have a data enclave, which we are building right now, where you keep the data locally, and then investigators are sending algorithms to the data, obviously in an enclave, which also security measures around it. The algorithm extracts the relevant information from individual patient-level information. This can contain PHI. But then the data is aggregated and exported as aggregate data. And the BDKD network has done that successfully across, I think, 4 or 5 databases in Europe, in 4 different countries. And this model really works if you have reasonably sophisticated IT infrastructure, people helping you with it. Clearly, we have them here in Michigan available if this becomes an aspect of relevance. But certainly from our vantage point, having rigorous consent, and at least in both rare and common diseases, our patients clearly state they want their data to be used. They are fully endorsing the open science, open community concept, and the experience with KPMP have anything short and accelerating. We have now systems biologists coming to the kidney field. Yes, we have a problem, but most of all, we have data where they can really solve hard problems. So if you manage that, if you get your community to learn and agree that everybody, including the scientists, including the junior faculty, wins when you do the transition to open science, it can be transformative.
Thank you. We probably have time for one more comment or question. All right, looks like everybody's hungry but wants to have their picture taken first. Right, Dorian?